Okay, so we've talked about the energy system so far with the phosphagen system, which is the ATP and PCR. Now, the next system that really comes online is the glycolysis system, and that's when we're going to metabolize or catabolize glucose. So let's talk about the metabolic pathways for this to happen. First, our very first step, of course, we have our glucose molecule. So glucose, and this is a 6-carbon molecule with a chemical formula C6H12O6. And first, you're going to phosphorylate it. So you would end up getting glucose 6-phosphate. And then another reaction and turn this to fructose 6-phosphate. And then we do another very important one here, and we get fructose 1,6-bisphosphate. And now, this is an important step. We're going to go through these. We get dihydroxyacetone phosphate, as well as glyceraldehyde 3-phosphate. Then we're going to kind of continue with this one here. And we'll put 1,3-bisphosphoglycerate. Now 3-phosphoglycerate. And another reaction here to get our 2-phosphoglycerate. And then we phosphoenolpyruvate, we'll just put PEP. And then pyruvate. Pyruvate, that's the end product of glycolysis. Okay, so let's talk about this pathway and the enzymes, some of the key regulatory enzymes that are important for this, all right? So when it comes to glucose, your first enzyme that's doing this, we call this hexokinase. And this is going to be in the muscle, specifically hexokinase 2. There's also hexokinase 4, which we call glucokinase. And that is in the liver. And in order to get this phosphate, we're adding the phosphate onto glucose, right? So we need a kinase. That's what we're doing with hexokinase. So this actually uses ATP here. So we have an ATP, and it gets converted to ADP, okay? And the enzyme that does that is hexokinase. And then let's talk about the hexokinase reactions for a little bit, though. In the liver and the muscles, a little bit different, because in the liver with glucokinase, that one is regulated a little bit differently, because it's the liver's job is to ensure that the blood always maintains enough glucose levels for the brain. And so that glucokinase is not going to be inhibited. For example, glucose 6-phosphate. If you have lots of glucose 6-phosphate, that can inhibit or decrease the activity of hexokinase, but it doesn't do that with glucokinase in the liver. And it's also regulated by hormones like glucagon, for example, in the liver, versus in the muscles, hexokinase is not regulated by that. So that's the first regulatory step. Now, also, it's important when you're adding this phosphate bond right here. That phosphate is anytime you're adding a phosphate, okay, it's important because it's going to trap. We can say it's going to trap the molecule in the cell. So let's say trap. So trap in the cell, because this way, with a phosphate, that's a large molecule basically, and it's charged. and it's going to prevent whatever molecule, in this case glucose, from leaving the cell. And that way the cell doesn't have to expend any energy to keep that glucose in there. Okay, so that's kind of one of the neat things about adding that phosphate in there. Once it's inside, it's trapped. It can't go back outside in the muscles. In the liver, there's another enzyme that it can do that. We'll talk about those in a little bit. Okay, so this is our first step here. Now we get glucose 6-phosphate, then it's going to just transfer into fructose 6-phosphate, and then we get the fructose 1,6-bisphosphate. Now, this key pathway right here is very important to add that other phosphate. You notice you have 2 phosphates on here now. So we also used ATP to get this. So we have ATP, and so this is an energy requiring step. And the enzyme that does this is PFK1. All right. Now, PFK1 or phosphofructokinase, this is a very key enzyme. It is a regulatory enzyme because this is the committed step of glycolysis. All right. So this is one you really want to understand, and we'll talk maybe more about that one here in just a little bit. But there's actually 2 enzymes we'll talk about: PFK1 and as well as PFK2. Okay, now we get to fructose 1,6-bisphosphate, and now we're going to cleave that molecule apart. Remember, so glucose is a 6-carbon molecule, and we're going to break it apart into 2 different 3-carbon molecules. One of them is dihydroxyacetone phosphate, and the other one is glyceraldehyde 3-phosphate. So we have these 2 molecules. Now, once the dihydroxyacetone phosphate is produced, there is another enzyme that simply converts the dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. But this is a reversible reaction, so it just depends on basically the mass action law, which direction it's going to go. Okay, so now we get our glyceraldehyde 3-phosphate. The next one is 1,3-bisphosphoglycerate. This reaction here is a very, very key one that we really want to understand, because this one we're going to form NADH. Okay, so we have NAD+, and it's going to get reduced to NADH. Now, notice though, what we're going to do here, there's actually 2 of these. There's actually 2 of these because the dihydroxyacetone phosphate was converted to glyceraldehyde 3-phosphate. There's 2 of these molecules. Therefore, there is 2 of these NAD molecules. There's 2 of them here. There's 2 NADH molecules here. Now, we also were able to add a phosphate because this molecule here is glyceraldehyde 3-phosphate, but we added another phosphate right now, so it's 1,3-bisphosphoglycerate. That didn't require ATP. We were able to just use inorganic phosphate. And of course, in this case, there'd be 2 inorganic phosphates because you're forming 2 of these molecules. We could just write 2 on all of these. So there's always going to be 2 of them. Okay. So we have 2 of these each molecules. Okay, so the enzyme that does this one is, we can write it right here, is glyceraldehyde 3-phosphate dehydrogenase. So just GAPDH. All right, so it's a dehydrogenase reaction, and so it's a redox reaction, oxidation reduction reaction, because we are taking electrons from the glyceraldehyde 3-phosphate and donating them to NAD+, and you're making your NADH molecules. And that's a very exergonic reaction, which is coupled with an intergonic reaction of adding the inorganic phosphates to the 1,3-bisphosphoglycerate. All right, now we have 1,3-bisphosphoglycerate, which is a very high energy molecule, and now we start getting our return on investment, per se, because here we can take ADP and we can convert it to ATP. All right, and actually there's going to be 2 of them because there's 2 of those 1,3-bisphosphoglycerate, and so that's a high energy molecule. So now we have 3-phosphoglycerate, and then that gets converted to 2-phosphoglycerate, and then you form by another rearrangement, and you get phosphoenol pyruvate, or PEP. Okay, and this is another one where you also are able to get ATP from. So we can do 2-ADP, and you get ATP. Okay, so you get 2 of those. So when it comes to this first part where you're adding those, sometimes you're using ATP, for example, like in this case, you're using ATP there. So this is the preparatory phase to start it off. Over here is the payoff phase, all right, where you're able to, what you get is called a substrate-level phosphorylation, all right? So we can just write here, substrate-level phosphorylation. And that's going to be contrast to oxidative phosphorylation. But there's a substrate-level phosphorylation. Just like creatine kinase is a very high energy molecule, or the creatine phosphate is a very high energy molecule, because the creatine phosphate has more, is a higher on the energy, that gives free energy, than ATP is. So the creatine phosphate can donate that phosphate group to ADP to make ATP. In the same way, these molecules here, 1,3-bisphosphoglycerate and 2-phospholipyruvate, those are high energy molecules. So they can donate a phosphate to the ADP molecules. And there's enzyme that does that. Like this one right here is pyruvate kinase, and you don't need to really know those for this. I just want you to understand some of the key regulatory steps. First, we talked about the hexokinase 1, and of course, glucose 6-phosphate. This one can feed back, and it can inhibit this enzyme, the hexokinase, but not so much the glucose 6-phosphate one. The PFK. Now, the phosphatidyl 1 is the committed regulatory step. Okay, so this is the committed. Like once it gets that level, or regulatory, once the glucose gets through this PFK1, it's gonna go the rest of the way. There's no turning back, right? That's why it's the regulatory step. And there's a number of things that can activate PFK1. And it should be logical. What do you think those things are? If you want to go through glycolysis and make your ATP, then the reason to do that is all based upon the bioenergetics. If you have low levels of ATP, in other words, if you have higher levels of, say, ADP and AMP, so things like our AMP, okay, even our ADP, okay, that's going to increase this level of phosphatidyl 1. There's another very important enzyme that can take this molecule and it'll make fructose 2,6-bisphosphate. Okay. Now this is actually called P. Let me write this a little bit bigger here. But this is the same idea. But this enzyme would be called PFK2. All right. And this fructose 2,6-bisphosphate is a very potent allosteric regulator of phosphatidyl 1. Okay, so we can write fructose 2,6-bisphosphate. And it's going to, all of these here are going to directly stimulate, all right, the activity of phosphatidyl 1. And then you can think some of the opposite effects are going to happen. So if you have, say, high levels of ATP or low levels of fructose 2,6-bisphosphate, I don't need to write so much here, but if you have high levels of ATP, low levels of fructose 2,6-bisphosphate, then that's going to decrease the PFK1 activity. If you have a lower PFK1 activity, then you're not going to run glycolysis. So this enzyme of PFK1 is a very key regulatory step in glycolysis, regulated by these main enzymes here. So if these levels are higher, they're going to upregulate PFK1, and if they're low, they're going to downregulate PFK1. And then there are other things. For example, if you have H plus ions, so if the pH is acidic, right? So a low pH, or if you have high levels of citrate, okay? So citrate, which we get into when we talk about the TCA cycle, all of these are going to decrease the activity of PFK1, okay? So there again, PFK1 is an enzyme that's allosterically regulated by a number of these key molecules. Okay, now going to back up here, there's another important thing I want you to understand. If we were to make ATP from glycogen, what would that look like? Well, let's say that we're going to take our glycogen. And this is a neat step because when you do this, we first make glucose 1-phosphate, and then this gets converted to glucose 6-phosphate. And in this step right here, this enzyme is called glycogen phosphorylase. And it uses an inorganic phosphate. So see, the glycogen is actually a bunch of glucose molecules that are attached to each other, and it makes these branches. And the higher out in the branches you go, the more the glucose molecules there are. So there's actually like tiers of these branches, and the further out you go, the more and more glucose molecules there are, and they're all attached to these non-reducing ends. And glycogen phosphorylase attaches the non-reducing ends of the glycogen molecule, and it's very favorable. When it does it, it uses this inorganic phosphate and does this like nucleophilic attack onto that portion, and it's able to add that inorganic phosphate onto the glucose 1-phosphate, and then that gets changed via phosphoglucomutase. Phosphoglucomutase to glucose 6-phosphate. And now notice though what the benefit that is. You're essentially missing, or you're skipping the use of an ATP. So hexokinase and glucokinase would normally use an ATP to get to glucose 6-phosphate. When you get it from glycogen, you don't have to do that. So that's one of the neat benefits of getting your ATP, I guess, from your glycogen, is you don't have to use one of your ATPs. Now, the liver, though, is also important, as I mentioned, to regulate your blood glucose levels. And so we might want to take our glucose 6-phosphate and put that into the bloodstream. And if in order to do this, we need to have an enzyme that can cleave off that glucose, or that phosphate. So we can say our blood glucose levels. So we want to go to blood glucose, and this is going to happen with a phosphatase enzyme. Okay, so now we have, this is an important point. Actually, to really talk about this a little more, because the fructose 6-phosphate mentioned is this regulatory step with glycolysis, and that's because everything turns into glucose 6-phosphate. The glucose 6-phosphate is kind of the key part that's going to regulate everything. And we can write it as your just G6P, and it's going to give us another options, right? So for example, you can store as glycogen, okay? So we get our glycogen storage, or maybe you can do blood glucose. You could also use some of these, these are still like basically broken down into TCA intermediates, and could be used as substrates for, say, amino acids even. So we just put amino acids. You can make these called glycans. Like, for example, the glycocalyx. Do you remember on your with your muscle cells, and you had the endomysium and the glycocalyx, right? And then you had the basement membrane. So that was that sugary layer. So we're talking about like and you have other things like your proteoglycans, all these sugar molecules. So this is also a regulator of or can be used to make all of those things. And then another big one that's very interesting is called the pentose phosphate pathway. And basically, just to give you an idea, you take this this glucose 6-phosphate molecule, and you're going to break it down to, you're going to create this ribulose 5-phosphate. And anyway, there's a, but in this process, a number of steps. But what I think is important to know is you would actually be taking your NADP molecules, and you're making NADPH. So we have like our NADHs over here, but NADPHs, that's more involved in your like anabolic reactions and for building things, fatty acid synthesis and so on. But even for as a reducing agent. So for example, you have this molecule of glutathione, and which serves as an antioxidant, and when it gets oxidized, then it needs to get replenished. So we can take, for example, your glutathione, and you would take it, so you have your oxidized glutathione here, and there'd actually be two of them on each one. So your NADPH can then reduce your oxidized glutathione to normal glutathione, and then that can go and continue doing its entire things. But that's the pentose phosphate pathway, and then of course this could you continue and you can make your nucleotides, for example, your nucleotides such as your DNA, right, RNA, and so on. And then of course the most important one is glycolysis, right? Glycolysis, which that's what we have right here. Okay, so my point is that glucose 6-phosphate is kind of the hub, of all these many other pathways that could be going on right here. But once you get through fructose 6-phosphate and go through this enzyme of PFK1, then that's when everything changes. That's a committed step, and you're going all the way down, and you get to pyruvate. Now, that's glycolysis. And pyruvate, though, there's different fates of what can happen to pyruvate they can happen along with glycolysis, but it's not the same as glycolysis. Okay, let's write those two things here. So, for example, pyruvate. Once we get down here, one of the things it can do is it can get converted to lactate. So in this case, we'll take our NADH that we formed in this reaction here, and it's going to convert it to NAD+. NAD+. And the enzyme that does this is lactate dehydrogenase. All right, and so when you're actually doing this reaction, you'll notice that it actually takes a hydrogen ion too. We'll talk more about the importance of this, but this is the most important thing about why this reaction is so important and so critical is in order for glycolysis. So if you have glycolysis that's running and you're trying to make as much ATP as possible, well, in order to continue driving glycolysis, you have to continue oxidizing glyceraldehyde 3-phosphate. In order to oxidize glyceraldehyde 3-phosphate, you need to have enough of the oxidized form of the NAD molecule. You need NAD+. If you were to reduce all of your NAD pluses to NADHs, you can no longer drive glycolysis. So as soon as this NADH is created, then it actually turns up right over here. Okay, I mean, these ones are like literally like right by each other actually, because this is all happening in the cytosols. They're cytosolic enzymes, and so you make 1,3-bisphosphoglycerate, you make NADH. NADH comes over here, and immediately by the action of lactate dehydrogenase, you're converting pyruvate to lactate. So it happens instantaneously, and a key aspect of that is that you can regenerate your NAD+ and continue getting this pathway going more and more. So then lactate production allows you to exercise more. It's actually what it both retards acidosis and slows down your fatigue. It prevents fatigue largely because it's able to regenerate your NAD+ level, so you can continue undergoing glycolysis and generate the ATP. Now, some people are here that lactate is produced under anaerobic conditions. Yes, that's somewhat true as well, but the real reason why lactate is being produced is because the rate that you need ATP is a lot faster than what you can get from the TCA cycle and the electron transport chain. So that's why we end up making lactate, so we can just do glycolysis, because you are getting your ATP in these substrate-level phosphorylations, and it happens a lot faster than going through the TCA cycle and then through the electron transport chain. And so it's the kinetics that drive lactate production, not oxygen availability. So when we look at the end products then of glycolysis for our ATP, let's just look at what we happened, right? So here we used an ATP molecule, okay? So we can just say that's a minus 2 ATP. Okay, so we use an ATP here and we use an ATP here. So that's minus 2 ATP. And then we get the payoff phase. Well, here we get 2 ATP. So here we get plus 2 ATP. And now we have this one right here, this plus 2 ATP. Plus 2 ATP. So your net ATP would be 2 ATP. But you also get your NADH molecules, okay? Looks like ATP, as well as NADH molecules, which will then go and deliver to electrons indirectly to the electron transport chain. It goes through this malate aspartate shuttle, which we'll talk about. And then you get more electrons from that pathway. Okay, so here's our, we're getting 2 ATP, 2 net ATP from glycolysis using glucose, but actually see if you use glycogen, you could potentially get, or 3, right? So you could potentially get 3 ATP if you used the glycogen. And this whole pathway is regulated by your bioenergetics, by thermodynamics, right? When you have low ATP levels, high AMP levels, AMP is going to drive phosphatidylinositol, right? It's also going to drive, of course, even this guy, right? This here, it'll drive, we get more blood glucose going out on the phosphatase enzyme. So all these enzymes are importantly regulated by substrates and by energy levels. All right. Now the other pathway of pyruvate. Well, then pyruvate eventually was going to go through what's called the PDH complex, and then go and participate in the TCA cycle. TCA cycle. So let's talk about that right here. So what does that look like? So we take pyruvate that's made. So we take pyruvate, and this is made as an end product like glycolysis, made in the cytosol, and now we're going to go into the mitochondria. All right, so maybe we can draw. We draw a little mitochondrial membrane here, okay? And that's the outer membrane. Then this would be the inner membrane. Okay. We'll have to come back and fix this a little bit, I think. But this pyruvate is going to enter into a specific pyruvate transporter. Actually use a hydrogen ion as well to get through. And it comes through this transporter. And the first thing it's going to do is going to meet this PDH complex. All right. Now, this is a very fascinating complex because it involves 5 different coenzymes. 4 of them are derived from vitamins, and it's tightly regulated, and it's one of the neatest enzymes that there are. All the things is there's 3 different enzymatic, 5 different reactions, but 3 different arms to it. So let's talk about how this PDH complex works. So first, pyruvate is a 3-carbon molecule, and so we can actually we're going to basically undergo a decarboxylation reaction. All right. So if we take this molecule of pyruvate, so let's go carbon and here and put here. Okay, CH3 here, and This is large. And now we're going to decarboxylate this, and we're going to add a CoA onto it as well. That I can just write. So we have like our CoAsh, just to put an emphasis on our sulfhydryl group that we're adding right here. Let's see, we have NAD being used. We have our TPP. This is thiamine pyrophosphate. So it has like thiamine, vitamin B1 in there. Lipoate is another one. And there's also FAD. So that's another vitamin, right? You have niacin, for example, and NAD and FAD. Let's see, you have, for example, vitamin B. Actually vitamin B5 is involved in this one right here for the CoAsh. But you have all these different vitamins in order to get this entire reaction to happen. And now you're just going to end up with a 2-carbon molecule. Okay, so you have your carbon, and now when you added that the CoAsh right here. Okay, actually maybe draw it a little bit different here. Here, and now you're going to get your CoA. Okay, and then your CH3, and plus the CO2 because that was decarboxylated. Now, okay, actually with this NAD, you're actually going to be getting an NADH from that. So the FADH₂ is involved, but you don't get electrons from there. Those electrons are then transferred onto here, and you get your NADH from this reaction. So I guess technically you get 2 NADHs because you have 2 of these pyruvates that are coming through this complex. Okay, now you finally get your acetyl-CoA. This is what you ultimately want to do. And now you can finally start doing the TCA cycle, all right? The tricarboxylic acid cycle, the citrate cycle. Okay, so let's start here. We'll take our oxaloacetate. And that's going to combine with our acetyl-CoA to form citrate. Then we get our isocitrate. Now we'll form our alpha ketoglutarate. And then we'll form our, let's see, about right here, succinyl-CoA. Succinyl-CoA. I'm just going to go through all these steps here, and then we can come back and talk about it. We get our succinate, and then our fumarate. Fumarate, and then our malate. And then oxaloacetate. So how does this happen? We take our oxaloacetate and we're going to convert it to citrate, and that's going to be because we'll take this acetyl-CoA right here. We'll just write acetyl-CoA, right? That's this guy here. And it's going to come in and combine with oxaloacetate, right? It's a 4-carbon molecule to make citrate a 6-carbon molecule. And it does so by this enzyme, citrate synthase. Okay, and now citrate is going to get changed a little bit to make isocitrate. And then the isocitrate will get changed to make alpha ketoglutarate. This is an important reaction because here you're actually going to do another decarboxylation. So you're actually going to lose a CO2 molecule here, and you're going to end up getting an NADH. Okay, so I just put NADH. So it contributes to an NADH molecule here because this is a dehydrogenase enzyme. So isocitrate dehydrogenase. Now, the very next step is also a key regulatory step. Taking alpha-ketoglutarate to succinyl-CoA. This also does the same thing, is it's going to give us a CO2, and it's the enzyme is simply called alpha-ketoglutarate dehydrogenase. Okay, because of the dehydrogenase reaction, what's going to happen? Look, you're getting another NADH, right? So now you have your succinyl-CoA, and now we're going to convert this to succinate. It's another important reaction because it's going to take, as it takes succinyl-CoA to succinate, you're creating a GTP molecule. And of course, GTP just gets transferred to ATP from a simple reaction, right? You have, for example, GTP plus ADP is a reversible reaction to make ATP plus GDP. And the delta G on that is like pretty much 0. Is equal to 0, prime naught. So it's just a reversible reaction. It just depends on the concentration of each one of those. Okay, so this enzyme is actually kind of cool too that does this. This one would be called the same thing as the name, succinyl-CoA synthetase. Yeah, the synthetase enzyme. Also, I guess another name for this is called thiokinase. But I like the synthetase because you are, again, you're doing a substrate-level phosphorylation. You're making GTP. Okay, now we have succinate. Now we're going to go to fumarate. And this is another important reaction, taking succinate to fumarate. It's interesting. All of these sides, like every single one of these first steps, are very important because they're contributing either electrons as in the form of NADH or a GTP, and this one's no different because in this reaction, you are literally converting, I'll draw it on this side, you are taking an FAD molecule and you're converting it to FADH. So that's, of course, we're going to be able to do some work with that in electron transport chain. This is actually the exact same enzyme. This enzyme is called succinate dehydrogenase that does this. So we'll just put it here. Succinate dehydrogenase. And now if I have fumarate, and then we're going to lose some water, and we're going to go to malate, and then malate to oxalic acetate. And this is another important one, because during this reaction, you're going to make another NADH because this enzyme is also a dehydrogenase reaction. It's malate dehydrogenase. Okay, and this is also important, this malate, because malate is able to get through the mitochondrial membrane. It's one of the few things that can do that. And so there's an important shuttle with the aspartate. So like you have like aspartate over here. You have aspartate and oxalic acetate and the alpha-ketoglutarate. And there's an important shuttle. We'll talk about it later. But there's an important shuttle for malate to be able to go back and forth and be able to carry the NADHs, because NADH is not permeable to this inner mitochondrial membrane. And so in order for those electrons from, say, glycolysis, in order to get into the TCA cycle, it has to go through this malate, aspartate, alpha-ketoglutarate shuttle mechanism. And you have malate dehydrogenase right here to make an oxalic acetate. So, actually, oxalic acetate this is one interesting thing is this concentration, the concentration of OA in the cell is actually really quite low, and it regulates a lot of these steps because if it's like super high, for example, you're going to have more citrate synthase activity, so you can get more citrate. So there's some key regulations that we want to talk about with each one of those. So if you also have like high acetyl-CoA, for example, if acetyl-CoA is high, then this is going to be an activator of citrate synthase. So citrate synthase is, I mean, that's the first step in this entire reaction. And so if you have high levels of oxaloacetate, high levels of acetyl-CoA, of course, these are substrates, so it's going to drive this reaction, so you get higher activation, right? But you have other activators, right, like ADP, for example, and that's going to activate it. And then now that's an important enzyme, but one of these other enzymes that are even more, I guess, stronger regulators are the dehydrogenase enzymes. So like isocitrate dehydrogenase, this is going to be upregulated by ADP and actually calcium as well. And that's also true for this one right here. So your alpha-ketoglutarate dehydrogenase, if this is high, and calcium, okay, all of these will upregulate this isocitrate dehydrogenase. And then you have your, other things like oxaloacetate, that could cause an upregulation of these. And then if you have, malate dehydrogenase is another one. But if of course, if you have higher levels of NAD+, right, because this is the reaction, so NAD+ levels, if those are high, then you could be driving this reaction, right? And then this works with the various inhibitors as well. So if you just think, again, based on the energy, if you have high levels of ATP, then you're going to want to slow down this whole cycle, right? So high levels of ATP or NADH or even citrate, for example, or succinyl-CoA, these ones will start to inhibit or slow down these types of these specific reactions. But I want to point out, though, how neat it is with calcium being an activator of isocitrate and alpha-ketoglutarate dehydrogenase. Because, again, when you're doing your exercise and what we know calcium is so important for your muscle contractions, right? It's going to be released from the sarcoplasmic reticulum and binds troponin, and it's there in the cytosol. It could be activating, the calpain system. It could be activating your AMPK pathway, by calcium calmodulin, but it's also activating your alpha-ketoglutarate and isocitrate enzymes and making this whole process much more active. And that's why when you actually can measure these enzymes to look for improvements in mitochondrial activity, right? Like what is the citrate synthase concentration activity, the alpha-ketoglutarate dehydrogenase activity. all of these are good markers of your mitochondrial function activity, and your calcium, it makes sense, right? It's going to regulate and activate these enzymes. Okay, so now with this cycle we just went through, what we just did was just with one acetyl-CoA. But remember, there's actually two acetyl-CoAs that are coming through because you had two pyruvates, right? And so I guess there'd be like a two pyruvates, two CO2s right there. But that means that if you have with these NADHs, you had 3 NADHs that happened, so it means you actually got 6 NADHs in total, right? So you have 6 of those NADHs, and then here you end up getting 2 FADHs, FADH₂s specifically. Okay. Now, and you get GTP, so you get 2 ATP levels, right? So if we talk about the products that you get from the TCA cycle, including the PDH complex, right? What do you end up getting? So NADH-wise, you end up getting, we counted 6 of those, FADH₂s, 2 FADH₂s, and you get 2 GTP, all right? So every because every acetyl-CoA goes through this, two times. So you end up getting 2 of these. So now the question is, what do you do with your NADHs? That we know with the ATP what you can do with that, right? It's energy, and so it can be shuttled out of the mitochondria, mainly through that creatine phosphate shuttle. But what do we do with these NADHs, and how does that give us energy? So now we need to go to the electron transport chain, because that's really where we drive our energy at. So let's see if we can draw that on this map here. I'll erase this, and I will do it right here. That's what I'll do. I'll erase part of this. Okay, so I just erased this portion because I want to draw the electron transport chain mechanism kind of right here. I want to try to do that. And this is the mitochondria, okay? This whole thing is, this is the inner mitochondrial membrane, okay? And here we're going to have our complexes where we're going to be able to use our NADH and FADH₂ molecules so that we can drive, finally, our ATP synthase enzymes. So our first one that we'll write will be our complex 1, okay? This is our complex 1. Then we're going to draw our complex 2 right here. Then we're going to draw our complex 3. Then we're going to draw a complex 4. And then we're going to draw our most important one, complex 5. Really it's just called their ATP synthase enzyme. And we're going to show how these, how this is the electron transport chain. So I'm just going to write ETC, right? Electron transport chain. All of these are. And we're going to show how you're going to make a bunch of ATP from doing this. Okay, now, oh, you know what? This is how we make our NADH, but we also can make other NADH and FADH₂s by doing beta-oxidation of fatty acids. So I want to actually come back to this, and I want to talk quickly about beta-oxidation of fatty acids. We're going to keep it pretty simple. Maybe we can just do it right here, for example. So we have a fatty acid that's in the, we want to oxidize it, right? So the very first thing that we need to do is, well, let me just draw one here. Okay, so let's go like this. And we're going to take, we'll have our basically our group. And we'll put, let's see here. All right, here. So this is, we're going to say this is, your typical 16 carbon. So like a palmitic acid, for example, 16 carbon fatty acid. And we want to oxidize this thing. Now, the first thing that has to happen is we have to add on this CoA. So you add on to acetyl-CoA, and that requires 2 ATP to do that, okay? Well, it actually requires, it actually takes, no, it's right here. It takes ATP, and you get AMP plus your, so it's your, so it is the different reaction is very exergonic, so it's very powerful because this requires a lot, and then this actually breaks up later. But this is equivalent to 2 ATP, right? So that's why we write it. So it's basically minus 2 ATP in this first step in order to oxidize the fatty acid. Now, fatty acids that are, if they're 12 carbons, okay, if they're 12 carbons or less, then the fatty acid actually can just go right through into the mitochondria, and it gets acetyl-CoA. It gets the CoA attached in the mitochondria. But if it's not, if it's longer than that, like if it's 14 carbons or longer, then it has to go through a specific shuttle. It's called the carnitine shuttle, basically. And so in order for it to do that, you first add this acetyl-CoA, and now we can add this membrane here. So you're going to take this CoA right here, and it's going to come through so you can make your fat. I'll show you here in just a little bit. So now, in order for it to get through, it has to go through this transporter, which involves a very important, it's called the carnitine transporter, basically. All right, so you've probably heard of carnitine before. I'm just going to put carnitine right here. Carnitine palmitoyltransferase. We can just put actually CP1 here, CP2 right there. And what's it's going to do is going to take acetyl-CoA and it's going to connect the carnitine palmitoyltransferase 1 will basically put a carnitine onto this fatty acid, and then it gets shuttled through the mitochondria, and then the CP2 will remove that carnitine and put the acetyl-CoA back on, and then it can undergo beta oxidation. Okay, now I say it because the CoA is too large to get through this inner mitochondrial membrane. It can't do it. So that's why it goes through this mechanism of this carnitine shuttle. So sometimes people will want to take like carnitine supplementation because they're thinking, hey, I'll take more carnitine so I can do more fatty acid oxidation. One of the problems with that, of course, if you look at this, the mechanism, we didn't go into detail here, but the carnitine is basically just going back and forth. It's recycled, right? You take acetyl-CoA out here, you put a carnitine on, it brings it through, the carnitine comes off, another CoA comes on, and then the carnitine comes back, and it just switches back and forth. So this is an anti-port mechanism that's just going back and forth. So for most people who are healthy, unless there's like, some sort of deficiency, a disease state or whatever, like carnitine supplementation probably isn't going to do very much, because the rate limiting step of fatty acid oxidation is not carnitine availability, right? I mean, this is a very important rate limiting step of getting it in there, but there's other things that other regulators. It's not the carnitine availability that's showing it down. There could be like malonyl-CoA, for example, and other substrates or other molecules, phosphorylation issues that are happening that's slowing it down. So, point is, we just want to get the fatty acid into the mitochondria, which again, if it's a longer chain fatty acid, has to go through this shuttle. It could be one of the benefits of taking a shorter chain fatty acid, because you do bypass this method and you can shuttle it right into the mitochondria, and you can do your TCA cycle, which this happens very, very quickly. But for beta oxidation, the reason why we call it beta oxidation anyway is because you take this carbon here, well, then you count. So this is the alpha carbon, and then this here is the beta carbon. And you can keep going to the end, right? Then you have your omega carbon. So in different places, like the peroxisomes area, they actually can undergo omega oxidation. But here we're talking about beta oxidation. And in beta oxidation, it's just this repeated cycle over and over again, where you're chopping the molecule in two parts every single time, and you end up just getting your acyl-CoAs. Okay. So there's just two carbon chains, two carbon molecules, right? Pretty much what you get right here, right? Your acetyl-CoA, and you chop it up in two, and then it comes right into the TCA cycle. So beta oxidation occurs in the mitochondrial matrix, and then the acetyl-CoAs that are produced go into the TCA cycle, and you do the exact same process that we just barely talked about, but with fatty acids instead of with the pyruvate that we talked about before. Okay, and when you do that, okay, there are, when you do chop this up, you end up making a lot of FADH₂s, and so you can actually take, for example, if we were using your palmitic acid example, for example, then in this case, during beta oxidation alone, you're going to end up getting 8 of your acetyl-CoAs, 8 acetyl-CoAs, and you would get 7. Oh, that's terrible, 7. but everything else is terrible. I apologize. So you end up getting 7 FADH₂s, and you get 7 NADH molecules. And then these will go to the electron transport chain, okay? We should actually capitalize this. Now it looks really bad. Okay, so now you have your electron transport chain. You just did a whole bunch of beta oxidation, so you actually have not only what you get from the TCA cycle, but you have all of these guys. Now, every acetyl-CoA that goes through here, you're going to get more NADHs. So we can calculate all that up here in just a little bit. But now let's talk about how do we actually use their FADH₂s and our NADHs through this process, right? Okay, one thing that we need to clarify here, because as I said, this is the mitochondria, this is the matrix of the mitochondria right here, and now here is a cytosol. Now it does become a little bit confusing because this membrane, the mitochondria, is not just one membrane, but there's actually two membranes. There's an outer membrane and an inner membrane. And right here was really this outer inner membrane. There's this whole aspect that I didn't really draw correctly. I was just trying to go fast, but it becomes more important when we realize that there's actually two membranes that are going along this whole thing, because right here we want to make a little bit of a divergence, and there's another membrane that's going to be over here. They're not connected this way, but basically the outer membrane is right here. Okay, this is the outer mitochondrial membrane, and then over here is the cytosol. So you have this other membrane that's right here. Okay. If I just keep on drawing here. All right, so there's two different membranes here, and here is the inner mitochondrial membrane. Okay, this is the inner mitochondrial membrane space specifically. All right, space. And this is important because this inner mitochondrial membrane is where all this activity of the ETC takes place. And I want to actually start with this complex right here. Okay, this is complex 4. We could just say this is complex 2, and this is complex 3, complex 4, we say complex 5. Because this reaction right here is actually going to be oxygen, and you're going to make water. H2O. Actually, I want to just do half of an oxygen molecule to make water, just for stoichiometric purposes. Oxygen is a strong oxidizer, okay? It's going to, it is known as the final electron acceptor. This is called the electron transport chain. So we're literally going to move electrons down this gradient in order to basically oxidize NADH and FADH₂s and end up creating water, all right? This entire process of glycolysis and everything is driven by oxygen, right? Because ultimately, we take glucose, C6H12O6, and we combine that with 6 molecules of oxygen, and we end up converting that to what? To 6 CO2 plus 6 H2O water molecules. And here's the water molecule right there, right? Water molecule, 6 CO2. We talked about the CO2 right here, CO2 right there. That's 4. And then over here, remember we had the acetyl-CoA that was produced. We could put here plus 2 CO2, right? There's your 6 CO2s. So we're literally oxidizing the entire glucose molecule. It's literally oxidation that's happening. And because by by decreasing the NADH molecules, because of this oxidation that's happening, that's driving the entire oxidation process from glucose all the way to water. We call it metabolic water. And the purpose of this is for this enzyme right here, this little nanomotor right here, and you could actually draw this a little bit differently even. But this enzyme is going to take your ADP and convert it to ATP. That's ultimately what we want to do. So this is, as you know, a very exergonic reaction. It requires a lot of energy to do that. So how do you couple this together? Like, how is it that consuming oxygen right here, oxygen as a final electron acceptor, going to drive the ATP production? And this is the core part of it right here. What happens is these NADH molecules that are floating around, they're going to come over here. So you have NADH, and they're going to donate their electrons, 2 electrons, and you make NAD+, and you get 2 electrons donations over here. And because this NADH complex is a stronger oxidant than NADH, so it's going to want those electrons. And now you have a carrier. We'll talk more about this later, but like CoQ10, for example, I've probably heard of that before, CoQ10. It's going to carry those electrons into complex 3. And when it does that, complex 3 gets those electrons, and then it carries those electrons. These electrons then get passed over to complex 4 through another protein, cytochrome C. And then you go to complex 4, cytochrome oxidase, where it consumes oxygen. Now, during this process of these electrons, this has a very high affinity for the electrons compared to this one. And then complex 3 has an even higher affinity than complex 1. So it's even a stronger oxidizer. So remember when we looked at the redox potential tables, you can actually look and see what specific cytochromes we're talking about. And those oxidation states are higher, and they're going to pull the electrons. And then finally, complex 4 is going to pull those electrons even more. And then oxygen is the strongest oxidizer. So it's going to really accept those electrons, and it's going to make water. During this process, it's going to do work because you also are taking hydrogen ions, okay? You're taking hydrogen ions, so H plus hydrogen ions, and you're literally pumping them to this complex, and you're going to pump 4 H plus ions in this complex, all right? And now we take complex 2 right here, and I'm going to rewrite this so I have a little bit more space. So we get NAD+. Here we take, this is where FADH is at, and it gets oxidized to FAD. Now, this is succinate dehydrogenase, right? This reaction right here, succinate dehydrogenase is doing this reaction. Well, this enzyme is actually the same enzyme as this one. So it's actually embedded in the membrane. So NADH is there floating around, but the succinate dehydrogenase enzyme, that is this enzyme. It's embedded in the membrane. So its complex 2 is called succinate dehydrogenase. And it's going to oxidize FADH2 to FAD, but there's no proton pumping that happens. And then these electrons are going to get passed also to CoQ10 and get to complex 3. And in this case, you're also going to be pumping your hydrogen ions, and you get 4 protons pumping right here. And now it's going to get to complex 4. And in this case, you are going to receive electrons, okay? So actually what happens is you take 2 electrons, right? These same electrons right here, you're going to get 2 electrons plus 2 hydrogen ions, okay, or protons. So 2 electrons plus your 2 hydrogen ions plus half of an oxygen molecule, okay, is going to make H2O, all All right, that's the stoichiometry that we're talking about, okay? Is 2 electrons plus 2 protons, right? Because there's 2 hydrogens right here on the water, right? There's 2 hydrogens in the water molecule, 2 hydrogens, 2 electrons, and a half of an oxygen molecule because there's only 1 oxygen, right? That's the stoichiometry that makes all this make sense. Let's just think about this again. These electrons come over here to complex 1, and complex 1 wants to take them away from NADH because it's a stronger oxidizer. And then CoQ10 takes those over to complex 3 because complex 3 is a stronger oxidizer. And then it lives in complex 4 because complex 4 is a stronger oxidizer. But then oxygen takes those electrons because oxygen is an even stronger oxidizer. So those electrons that were dumped over here end up going all the way to complex 4, meet up with oxygen, and when it does, it gets those electrons, it combines with 2 protons, so essentially you're essentially pumping 2 protons, right? So we can just say this is equivalent to 2 hydrogen ions right here. Okay, this will come in handy in just a little bit. And then you make water as the final product. So you get a water, and then of course you get your CO2, and that's why you have this equation right here, that glucose plus 6 O2s goes to 6 CO2 and 6 water molecules. So you'll see all the stoichiometry will map out perfectly. Now, why is this important? Why is pumping these protons so important, right? So you have a very high gradient of positive charge right here. Well, the reason is because these hydrogen ions are going to want to do what? They want to come through here, okay? They want to come through and come back into the matrix. This is the gradient. So you have a delta in the pH, all right, is what you're really making. So a delta in the pH gradient is like 0.75, or it could be higher even. So the mitochondrial pH, the pH of the mitochondria could be, it can be close to 8, for example. Probably depends. And then the cytosol, depending on the exercise and things, it could be lower, right? It could be the cytosol over here, pH 7, right? Or even lower, like maybe even 6.5. So there's actually a number of factors that determine what this gradient is. But that gradient is very strong. And it's going to really make it favorable for the hydrogen ions to want to come back in here. And this little nanomotor, this ATP synthase enzyme is what it's called, as that ATP comes through, it's going to twist the nanomotor, okay, like this. And every ATP molecule, or ion, it's not, every ATP molecule that's created, okay, requires a total of 3 hydrogen ions for that to turn. And so ADP and ADP, and I should add plus a phosphate right here. Okay, these two right here are literally sticking on the ATP nanomotor. So the ADP and PI are like right there, and the hydrogen ion comes through and it turns them closer and closer and closer to each other until they finally connect. And once they turn enough, then the ATP gets kicked off, and now it can be used as energy. Now, so it takes like these 3 turns. But we also have to bring in the ATP and ADP, right? So you actually have another enzyme we could just do right here. Remember, this is the whole, you actually have 2, you have 2 membranes, right, that are coming through right here. But we'll just say this is like includes all of them. So maybe we could just go like this, for example. Maybe that's a quick way to trick the system. What happens then is we have ADP that we made out here, and the ADP is going to be coming in, and we have our ATP out here, and the ATP we want to come out, right? So we have, this is the dinucleotide translocator, right? And we also, there's another, we also need to have our inorganic phosphate. So we have our PI, and we also have a hydrogen ion, and these are going to be basically an antiport situation in order to bring the inorganic phosphate into the matrix of the mitochondria. Okay, so this is an important mechanism in order to get that ATP out and bring the ADP in. And ADP strongly drives ATP synthase, that enzyme, right, driving cellular respiration. So, because we're also using a hydrogen ion to bring in that inorganic phosphate and this process, we can say that in order to make one ATP molecule, so an ATP molecule is going to need 4 H plus ions, okay? 4 H plus ions. This looks, I want to make sure you can read this. We're going to refer back to it. This is just, so 4 H plus ions, okay? Again, 3 of them are going to be turning, the 1 is going to be used over here. So now we can figure out how many ATP is each one of these reducing agents worth, okay? So let's look at this. So with NADH, we have the first complex, complex 1, NADH dehydrogenase. What happens? We dump the electron, the 2 electrons here, and how many electrons does it pump? 4, right? So that's 1 ATP. It skips complex 2. Complex 2 does no proton pumping. It goes to complex 3. Now we pump 4 more protons. So that's another ATP. That's 2. And then we go to complex 4. We consume 2 hydrogen ions in this equation right here. So that's like 2 protons that are being pumped. So that's 6 hydrogen ions. Therefore, 1 NADH is worth how much, right? So an NADH is equal to 2.5 ATP. You see that? That's how we know the math on this, because of the electrons and protons being transported. So let's do the same thing for FADH₂. FADH₂, where is it getting oxidized at? Well, it's missing complex 1. It gets oxidized at complex 2. Well, complex 2 doesn't do any proton pumping, so it misses all of those. But you do go to complex 3, there's 4 hydrogen ions, so that's 1 ATP. It goes to complex 4, and so again, this reaction consumes 2 hydrogen ions, so that's about 6 protons. Therefore, FADH₂ is equivalent to how many ATP? That's right, 1.5. So FADH₂ would be equal to 1.5 ATP. Now, I know some texts will round this up to 3 ATP, and this will be 2 ATP. And it's this phosphate-oxygen ratio, and there's a lot of things that go into this, but this is the best account that I'm aware of anyway, of how to calculate how many ATPs are made per NADH FADH₂ molecule, and the stoichiometry works out quite well for this. Okay, so NADH makes 2.5 ATP. Because you're getting 6 protons, we could just go, right, 6 hydrogen ions, right? Versus this one right here is only doing, this one was doing, we said 4, 8, 9, 10, so it would be 10 hydrogen ions. But this one is where we're only doing 6 hydrogen ions. And that's why you only get that many. So now we can actually calculate how many molecules, or how many ATP molecules we should get from doing a complete oxidation of glucose, right? So we already talked about here from glucose we get our 2 ATP and NADH, and this should be 2, right? So we get 2 of our NADHs. So we can add all these up together for glycolysis. Maybe we can do that right here, for example. So in glycolysis, we get our 2 net ATP, and then we have our NADHs, and if we count all of those up, so we get 6 NADHs from all the pyruvate that's going on, plus we have these ones, the 7, 8, and then we have these ones, so that's 10 NADHs. Okay, so 10 NADHs, and that's going to be equal to how many ATP? Well, we just go 10 times 2.5, right? It gives us 25 ATP. And then we have how many FADH₂s? We get 2 of those, right? So 2 FADH₂. We get 2 of those, and then 2 times 1.5 gives us 3 ATP. And then, of course, we have GTP, right? So we have, we made 2 GTP. So 2 GTP, which is equivalent to 2 ATP. And so now we just add these up and we get 25, 26, 27, 28, 29, 30. And 32, right? So we get a total is equal to 32 ATP. Okay. Now there's another consideration. Remember, if we go from glycogen, right? It could actually be 33 if we go from glycogen. And it's one more interesting aspect is it depends on what we do with these NADH molecules over here, because there's one other pathway that we haven't quite talked about yet. Well, actually, if we remember, we talked about this right here, this aspartate, alpha-ketoglutarate, oxaloacetate, malate shuttle, right? This is how we get these NADHs over here. How do we get them to go over here? And we can use this shuttle, right? Where we're going to take, for example, our malate, and we can put on their NADHs. Okay, NADHs to make NAD plus, and then malate can go through here using alpha-ketoglutarate shuttle. There's a whole shuttle here. We're not going into the whole thing, but now we can get the 2 electrons finally into here, and then the malate gonna go is reaction with oxaloacetate or something, and you make your NADH levels, which contribute to right here. So this gives you 4 total proton pumping. Now, there's another mechanism that so different tissues use different mechanisms. Okay, there's another one called the glycerol dehydrogenase shuttle, or glycerol 3-phosphate dehydrogenase. Maybe I'll put it just over here, just because it's what I really want to show. But this is this glycerol 3-phosphate shuttle. And what happens is the electrons are actually donated right into complex 2. So if you donate these electrons to complex 2, you end up missing your complex 1. And if you miss your complex 1, you're not getting your 4 protons. So when you oxidize glucose, you end up only getting 30 ATP. All right, that's why we typically write it's 30 or 32. Okay, that's like 3,000, but 30 or 32 ATP, because it depends if you're going through the malate, aspartate, alpha-ketoglutarate shuttle, or if you're dropping with the glycerol 3-phosphate shuttle. And it depends on the tissues. Okay, so when it comes to the brain and muscles, you're actually using this shuttle system right here. So in the muscles, you use this shuttle system versus the heart and other tissues, you use this shuttle. And so those other tissues, you end up getting more ATP. But you don't need to get glycolysis as quickly, or the ATP as quickly. So maybe that's one of the differences that are going on. So the count of ATP really depends on if you're using the glycerol 3-phosphate shuttle or if you're using the malate alpha-ketoglutarate aspartate shuttle. And that accounts for the discrepancy of how you're going to count them. The other part that I wanted to touch on before, we've talked about this already with the creatine kinases enzymes, because remember over here we have the, you have your sarcomere, you have your sarcomeres. Let's just draw this right here. And you have your creatine kinase enzyme right here on the M-line protein, and you're using a whole bunch of you're doing ATP hydrolysis. So we can just do ADP, right? And you make your ATP plus creatine. So I can do creatine phosphate. Right, this is happening very quickly next to your myosin ATPase, your circuit pumps, and everything like that. Well, that's happening right here in the cytosol, along with glycolysis and everything else that's happening. But we're doing, we're making a ton of ATP, as you can see, right? Like most of your ATP comes from inside of the mitochondria. How do we get the ATP from over here all the way to over here? Well, we talked about this translocator, this dinucleotide translocator, and it's going to diffuse over there, but it still takes time for that to happen. So actually, there is another enzyme just in this space. Okay, this is the creatine kinase space in the inner membrane space, basically. Because again, if we do this membrane differently, okay, you would actually take this ADP that comes out, and you would combine it with creatine, and of course, you know the drill. You make creatine phosphate plus ADP. And the creatine phosphate can then come over here and be used right here. And then this ADP over here would just come right back in, okay, and drive your ATP synthase. Because like I said, high levels of ADP, they drive the ATP synthase a lot. So this is a very elegant system because ATP is a large molecule. Even though it's only 3 letters, it's actually a large molecule, and it has to diffuse all the way over here, and it's going to take some time to do that. But creatine phosphate is a smaller molecule, and it's faster to diffuse. So we can think of creatine phosphate as kind of the energy carrier versus ATP is used like locally there for energy. So this energetically, it's favorable for the reaction to proceed this way from the creatine kinase on the mitochondria. And this is really what couples oxidative phosphorylation to what's going on in a cytosolic area with the sarcomere contractions and circuit pumps and so on. So creatine phosphate is created here, delivers over there, you make your ATP, creatine, and can circulate back over here. ADP enters back in, right? It comes out and gets dephosphorylated, goes right back in, and you're able to really couple this energy so it becomes very efficient, right? Now, we've talked about what happens to the oxidation of glucose all the way to make an ATP. I guess we can compare this to your fatty acids. We already showed our beta-oxidation that's happening here. So we could go ahead and calculate out how many ATP are we getting from this reaction here. All right, so let's go ahead and do that. So here I already wrote down we get 7 FADH₂s, 7 NADHs from beta oxidation, right? And then the acetyl-CoAs are going to go through here. And every single time they make it around, they're going to give you 3 NADHs and an FADH₂. And so we can multiply that out, and we can get a total amount of FADH₂s and NADHs, all right? So let's go ahead and do that, and then we can calculate the total amount of ATP that we get. So we can write the total here. So, okay, so we have 8 acetyl-CoAs. We go around the citric acid cycle, and we count how many times we're going to get NADHs. And then we add that to, so if we do our NADHs first, we have 7 NADHs. And so we're going to get a total then of, let's see, 31 of our NADHs. Okay, and then we'll have a total of 15 FADH₂s. And we'll have a total of 8 GTP. So this was going to give us 77.5 ATP, right? Because 31 times the 2.5 will give you 77.5. So 77.5 And then the FADH₂, so same thing, so 15 times over here, our 1.5, is going to give you 22.5. And then our 8 GTPs will just give you 8 ATPs. And now we total all those up, and we would get 108. 108 ATP. Oh, but you know, we talked about this right here. We minus ATP. We have essentially 2, right? So we actually minus 2 here. And so really, we would get a net of 106 ATP per palmitic acid. So we'd have, in fact, if we took our palmitic acid, they're 16 carbons long. Did I write that somewhere? No, maybe not. So we would have we could write this equation, for example, of oxidation of palmitic acid. So C16H32O2 plus so many molecules of oxygen, and that's going to go to, well, we're oxidizing, just like with CO2, we're going to be oxidizing that to CO2. There's 16 carbons, so you get 16 CO2, and of course, some water. Well, we make water right here, so we can actually, this is actually a good way we can actually check of how many electrons we actually need in order to make water. So if we do the stoichiometry, right? So for per oxygen molecule, basically, because right here, right, we have a half of an O2. So we could write this differently. We could do 4 electrons plus 4 protons plus O2, right, will give us your H2O molecule. And then we can, because that's a full O2 molecule, because we want to know, well, how many molecules of oxygen are needed in order to oxidize this. And so we can actually use this equation, and we can calculate based upon the amount of the number of electrons that are going through the electron transport chain to determine the stoichiometry to oxidize palmitic acid, to see how many molecules of water were formed and how many molecules of oxygen were formed, right? Okay, so let's use this equation to kind of guide this reaction, okay? And then we know how many electrons were extracted because we just did this math right here of how many electrons were used in order to or were taken out with these reducing equivalents. Okay, so let's look at this. So remember, with NADH, every one of these is 2 electrons, right? So you can simply multiply both of these by 2, and you get the total number of electrons. So 31 times 2 is going to give you 62 electrons, and then 15 times 2 would give you 30 electrons, and add that together, and you're going to get 92 electrons. That's how many electrons are needed. Okay, so you get 92 electrons. Now, using this equation over here, you have 4 electrons per oxygen molecule, right? So we can take 92 and divide that by 4 electrons. Okay, and as you do that, 92 divided by 4 is going to give you 23 oxygen molecules. So 23 then would go right here. All right, and then we know you'd actually be producing, because you produce water molecules on this side. So you would actually be making, because you have 4 protons, so you would actually be making 46 water molecules. Okay, so we can put plus 46 H2O molecules. And of course, in order to balance this side, then you'd have to add more water molecules. about 30 H2O molecules. But just because in order to keep the balance with the amount of hydrogens you have over here to just have a balanced equation. My point with this is we're literally able to follow every electron from the beta oxidation all the way till we get through the electron transport chain until we're making the water molecules. And we know stoichiometrically, as well as even analyzing with gas analysis, how much oxygen is literally consumed upon the oxidation of each mole of palmitic acid. And this is the equation that we end up getting. So we can do the exact same thing for the glucose, right? So we know we already wrote the equation over here, but let's confirm. How do we know it's really 6 oxygens? Besides, that's what the not even the equation, but the stoichiometry would suggest, right? So we go C6H12O6 plus we have O2 molecules, and then we're going to make our we get 6 CO2 plus 6 H2O. So do the same thing. So we add up how many NADHs and FADHs do we have in total right here, right? So we just barely did 10, so 11, 12. So we have 12 NADHs right there. And then we go 12 times 2 would be 24 electrons. So we can do the same thing. So we go 24 electrons. We divide that by 4 electrons. And what do we get? 6 O2. So that means that we need 6 oxygen molecules in order to extract the 24 electrons from glucose. And therefore, this would complete the equation. And it works because, look, you have 6 oxygen oxygens in the glucose plus these oxygen molecules here. Okay, and now you end up making, right, there's 6, so that's going to be 12 oxygens plus 6 and 12. So you end up balancing, right, every oxygen, every carbon, every hydrogen is all balanced out by this equation. Again, showing we can stoichiometrically follow every electron, and we can do the same thing for every proton. In fact, we're going to do that maybe later and show all the hydrogens that are accounted for, and you're going to find out that through metabolism, the breakdown of the catabolism of glucose doesn't really contribute to hydrogen ions at all. If anything, it could be argued it's the other way around, that catabolism does not contribute hydrogen ions in the way that we're often led to believe in making everything really acidic. It's other things that are going on. So with this understanding, you're able to really understand the whole bioenergetics, that everything, all metabolism is not some, I guess, magical thing, but just following normal thermodynamics, because oxygen as a final electron acceptor is driving all of this. And we can trace every electron, every hydrogen ion, all the way down through every pathway and account for everything. There's no hand waving that's going on here. Now, we did talk about how the ratio was like 30-32. That's if everything is 100% efficient, right? There's a little bit other interesting things that are going on that make that ratio go a little bit down, because you have another protein, for example. We can just put a protein in right here, all right? Even though it's like up over here, and okay, maybe I'll put it over there. We'll just leave that there because that's the transporter. We can put one in right here. This right here is this is called the uncoupling protein, okay, or thermogenin. We can't read that. Hopefully you're listening. Thermogenin. This is the uncoupling protein, because what happens is it allows hydrogen ions to just come right into the matrix. And that's waste energy. Right? You did all of this work to pump the hydrogen ions over here, but then you just allow hydrogen ions to go right in here. So you're not making any ATP when you do that. You just make a lot of heat. That thermogenin, or the uncoupling protein, is rich in mitochondria of brown fat. And that's how babies are able to stay warm. And there's a lot of talk about how brown fat is actually healthy because it helps with your entire metabolism, because it helps with energy expenditure. And there's a lot of really interesting research about the benefits of brown fat. And that's some of the ideas behind getting cold adapted and cold plunges is maybe you can increase the amount of browning. Yeah, there's some evidence for that a little bit, but it's not so impressive. It probably doesn't work. It's much better just to exercise if you want to get the benefits. But that's the idea. And this thermogenesis uncoupling protein. So one thing to think about is if you have a really high efficient metabolism, then every electron and everything you break down goes through perfectly, and you're able to make this 30 or 32 ATP from glucose, and you make ATP. If you have a so-called inefficient metabolism because you have a lot of, say, thermogenesis uncoupling protein, then you are burning a lot of energy and it's just going off as heat, and you're not producing a lot of ATP. And so there's a little bit of irony or contradiction, because some people, they look at somebody who seems to eat all the time and yet they don't ever gain any weight, and we say, oh, their metabolism is just so efficient. It would be the opposite, right? If anything, they have an inefficient metabolism because they have maybe more of this uncoupling protein, and they're not gaining as much ATP. There's actually other things going on. They're just expending energy more. There's the thermodynamics, the calories in, calories out aspect of it. Of course, not every calorie is the same way, which is we can actually discuss the reasons why when you are metabolizing, say, proteins, for example, versus fats or carbohydrates. Even these ratios for fats and carbohydrates are quite different for each other, which is why some macronutrients deliver a different amount of ATP, or provide different amounts of ATP compared to how much oxygen is being consumed, right? That ratio, or we can say like phosphate oxygen ratio. It's all fits in here so that we can understand the effects of foods and calories on our metabolism. So that's one interesting part with the thermogen, and I just wanted to add in there. And then this electron transport chain, probably we'll talk more about this so you can understand how critical this is in other areas, because, oxygen, as we talked about here, is being consumed as a final electron acceptor, but sometimes oxygen might get consumed over here in complex 1. And if that happens prematurely, it receives an electron, then what happens is you end up making this guy right here, the superoxide anion radical. And the superoxide radical can exert some benefits, but can also be problematic if it gets too high. And that's why you have enzymes like glutathione peroxidase that can, or superoxide dismutase specifically for this anion, and it can convert it to, say, hydrogen peroxide. And then you start using your other reducing equivalents like your glutathione levels so that you can combat that oxidative stress. Well, that's why we had this pathway over here. Remember we talked about your glutathione levels and the oxidized glutathione? That this is being made so that you can handle some of the reactive oxygen species that are occurring primarily around complex 1, maybe complex 3 of the mitochondria, and getting the entire system into homeostasis. Right, so we might spend a little bit more time on that and the importance of this entire process. Also, complex 3, this contains that very important protein, the RISP protein. Okay, this is the risque iron sulfur protein, which is which basically accepts an electron from ubiquinol or the CoQ10 and pass an electron one way. We'll go into the Q cycle later. But this is the protein where we published the paper in Redox Biology showing that hydrogen gas seems to target that specific protein and induces this downregulation. Well, think about what's going to happen if you don't have that RISC protein, is not functioning very well. You're going to have electron backups. You might get some superoxide levels, which we saw some of the increase in superoxide production, and that's going to stress the mitochondria. If that stresses the mitochondria, then the mitochondria needs to react, and it ends up increasing the amount of RISP levels, and then you get higher levels, or you return your baseline levels of ATP, so you have an improved function of the mitochondria. So it's just this dynamics, and that's kind of a hormetic-like effect on the mitochondria. So many things do do that. Many things sort of stress the mitochondria, and that in turn makes the mitochondria more a higher resilience to stress and improve its function. And also, when you are exercising, you are breaking down your ATP a lot and getting AMP, and then AMP, as we talked about, activates AMPK, and then AMPK activates PGC1 alpha, and then PGC1 alpha increases this whole thing of mitochondrial biogenesis, and markers that we can look at for like citrate synthase, alpha-ketoglutarate, and other of these key enzymes, so we can see how is oxidative phosphorylation, TCA cycle, and all of these things going. Okay, so this is just kind of an overview of everything. We'll take some of these and go into a little bit more mechanistic detail, and then we also want to talk about the lactate aspect and counting all of the hydrogen ions and all the protons, so you can see that the net effect of catabolizing, say, glucose, if anything, is slightly alkalizing or neutral, but it's not acidifying like we often think about. So, all right, thank you very much.